Hello everyone, my name is Nihal Hazem and I am representing the Egyptian Drug Authority together with my colleagues, Dr. Rania Hassan, Dr. Rania Shusha, and Dr. Asma Fouad. It's a pleasure to be speaking with you today. As regulators, our mission is to ensure patients have access to safe, effective, and high quality medicines while supporting sustainability of healthcare systems. So, in this presentation, we will share insights on the regulation of biological medicines and biosimilars, mainly focusing on how these tools can improve access to life-saving therapies such as Kanjinti, Trastuzumab biosimilar of Herceptin. Many of the most important medicines in the modern healthcare system are biological medicines, and one of these examples is Trastuzumab, a drug that has transformed the treatment of HER2-positive breast cancer. These medicines are so impactful that they are included in WHO model list of essential medicines. This list identifies medicines considered critical for meeting the most important health needs of populations worldwide because they provide survival benefits. But due to their high cost, has historically limited the patient access, particularly in public health system. For this reason, biosimilars play a critical role in overcoming these barriers. They are not just lowering the cost for their alternatives, they are also a critical regulatory tool to ensure timely and equitable access to essential biological medicines while maintaining high standards of quality, safety, and efficacy. For us as regulators, particularly in middle income countries, biosimilar pathways must balance between patient protection and public health needs. They do this through a stepwise comparability exercise, relying on the totality of evidence rather than unnecessary duplication of a large clinical trials. By, by doing this approach, this accelerates the patient access and support the sustainability by containing access and allow authorities to exercise scientific judgment and international reliance mechanisms all without compromising regulatory rigor. Trastuzumab represents a clear example for biosimilar regulation because its high clinical value and ethical consequences of limited access highlight why ensuring broad availability is critical. As one of the first effective HER2 targeted monoclonal antibody, Trastuzumab has transformed the prognosis of HER2 positive breast cancer both in early stage and metastatic settings, and it's also benefit with metastatic gastric cancer, conditions historically associated with poor outcomes, and also for HER2 overexpressions that occurs in approximately 20 to 30% of breast cancer patients, which associate with aggressive disease if left untreated. Herceptin, the reference trastuzumab product, is recognized by both authorities, WHO and EMA, as a cornerstone therapy. It has consistently demonstrated a clinical meaningful improvement in the overall and disease-free survival when administered either IV or subcut. These performances make it serve as benchmark for biosimilar comparability assessment, allowing assessors to determine whether the biosimilar approval ensure comparable clinical outcomes while supporting broader patient access. As illustrated in this diagram, when using Herceptin, we can see for the overall survival benefit for 5 years. In Egypt, trastuzumab was first introduced in October 2018 with the approval of Herceptin, marking a major advance in treatment of HER2-positive breast cancer following the global wave of biosimilar approvals between 2017 and 2019 in Europe and US. Several biosimilars products as Herzuma, Hertase, Ogavri, Enhertu, and Herticade entered the Egyptian market between 2019 and 2022 for local regulations by Egyptian drug authorities. Despite their availability, reimbursement, and consistent access remained challenging due to the cost and healthcare system constraints. This situation underscores the potential role of biosimilars in improving affordability and equitable access of HER2-positive breast cancer's targeted therapies. Now let's look at the clinical impact and benefits of trastuzumab. In early stage HER2 breast cancers, adding trastuzumab to standard adjuvant chemotherapy can reduce the risk of breast cancer recurrence, And this can be shown when make a large meta-analysis study of 13,864 women for a 7 randomized trials demonstrated that adding trastuzumab lowered the recurrence rate by approximately 34% and also significantly lowered the breast cancer mortality. And it's also improved the pathological complete response rate in the neoadjuvant settings. And it also illustrated in this diagram how when adding trastuzumab to chemotherapy, it showed that 12% absolute benefit of disease-free survival around 3 years. In the metastatic HER2 breast cancer, adding trastuzumab to chemotherapy improved the disease control and the survival rates. The clinical trial shows that the overall response increased from around 32 to 50%, while the median progressive free survival extended from 5 to 8 months, while the overall survival rates increased from 20 months to 26 months. Despite the metastatic disease is not curative, trastuzumab provides substantial survival advantages and better disease management, establishing it as a standard first-line therapy with careful cardiac monitoring due to potential cardiotoxicity.
Hi, how are you all? My name is Rani Ibrahim. I am General Manager of Clinical Trial in Egyptian Drug Authority. I am representing part 2 related to quality assessment. Today we are moving to part 2 for quality assessment for APP 980, which is known commercially as Kanjanti. As newly requirements, it's related to the approval of biosimilar, that the product is highly similar to the reference medicine. In this case, which is Herceptin, APP 980 is produced using a mammalian Chinese hamster ovarian cell lines. Our goal here is to demonstrate that the physical and chemical characterization of this biosimilar is. Now we talk about the quality aspects related to physical and chemical data of ABB 980, which is the primary structure content of amino acid sequence of APP 980 is totally identical for uh, Herceptin, the reference product, while the only difference here is related to the absence of C-terminal lysine, which is commonly occurrence in biotechnology as it is some variability, but it is considered to be clinically not relevant. That is, the disulfide bonding pattern and overall molecule mass are consistent of uh, consisting with the reference product regarding the glycolization, which is critical for biological function. The N-linked glycosylation at ASN 300 has been fully characterized. We found that glycoform distribution and all galactosylation and fucosylation is compared to Herceptin. Also, for the charge variant and isoform, there are minor differences observed, but within acceptable variability, no impact on the binding affinity or the pharmacokinetics. For the manufacturing and process control, APP 980 active substance is confirming that it's totally produced according to current GMP requirements at Python Biological in the Netherlands, which following the system uses two-titer cell bank system, master cell bank and working cell bank. The advantageous agents assays tests results indicate that the cell bank is sterile and free of any detectable mycoplasma and viruses, and also the manufacturing ensuring that robust and consistent process which is essential for maintaining the narrow quality ranges required by regulators as like EMA. The critical quality attributes, it is very important attribute for the biosimilar, oncological biosimilar. Generally, according to guidelines, its primary structure, amino acid sequence, glycosylation profile, change, charge variant, and isoform. But here, why this critical quality attributes is important, especially for oncology biosimilar or a monoclonal antibodies. It will focus on the attributes impacting efficacy, safety, and primarily biological activity. Like for efficacy, antigen DCC and HER2 binding directly related to how well trastuzumab fight cancer, or for safety, we monitor purity and impurities. Any significant in any significant full protein aggregation or host cell protein could lead to increased immunogenicity or toxicity, potentially causing infusion reaction or cardiac issue. So, by matching the quality related attributes for Herceptin, we prove that the biosimilar is both safe and effective. Regulatory framework use tiered approach, where tier 1 for the highest impact quality attributes, for tier 2 is for moderate, and tier 3 for uh, low, to define acceptable range. For this pathway, we prefer that we found that critical quality attributes for trastuzumab for biological activity is the highest impact, for HER2 binding affinity, with ability to bind to HER2 receptor on cancer cell, antibody dependent cellular cytotoxicity, also trigger immunocell to kill cancer cells strongly influenced by glycosylation, anti-proliferation activity, inhibitor of cancer cell growth. Also for glycosylation profile and physicochemical properties are for tier 2, which will be moderate impact, and also can affect the affinity, antibody dependent cellular cytotoxicity for binding, or by technique, either for fragment and high level chain host cell protein, which can be affected in case of impurities. Here we talk about the final assessment for trastuzumab Kanjanti product. Analytical similarity assessment is a must for the approval for any biosimilar product. Uh, the primary structure, glycan, map, and high order structure were all found to be similar. Most important were all found to biological activity assay showed no differences in the HER2 inhibition or antibody DCC activity, although there was slightly higher average relative to C1q binding in APP 980 that was not deemed clinically significant. For EMA conclusion, for approval, the comprehensive analytical similarity assessment, including comparative evaluation of biological activity, primary structure, overall, it can be concluded that from the quality point of view, that APP 980 can be considered a highly similar to Herceptin. In addition, the data was obtained showed that APP 980 for all relevant quality attributes are within narrow range of variability. Furthermore, the data show that the ranges obtained for APP 980 are very narrow, suggesting that the manufacturing process is under robust, consistent, and well-controlled process. As a conclusion, Kanjanti was approved because comprehensive comparability study across all quality critical attributes demonstrated high similarity to Herceptin, with minor analytical differences which were justified and not clinically meaningful. Therefore, it confirmed equivalent biological activity. also, we concluded that Kanjanti biosimilar would deliver the same clinical benefit risk profile as the reference product. Thank you all, and now forward to my colleague for part three.
I'm Dr. Rania Shusha, Manager of Protocols and Technical Studies for UP at Egyptian Drug Authority. I'm now talking about clinical pharmacology data that explain the comparative pharmacokinetic data for Kanjinti as a reference product versus reference Trastuzumab Herceptin EU. This is a comparative study demonstrate high similarity with geometric means ratio for K parameters like AOC, infinity, AOC last, and Cmax, showing by using validated ECL assay strongly support biosimilarity without immunogenicity impact on the exposure of monoclonal antibodies. This data assessed using validated ECLIA assay with high sensitivity, low variability, with no matrix effects across diverse demographic mirroring targeting population. Minor consideration regarding this study showing slight Cmax asymmetry, which reflect typical monoclonal antibody pharmacokinetic variability, but remain non-inferior within confidence interval. So overall, this data obtained from pharmacokinetic similarity indicate confirm comparable exposure profile warranting regulatory reliance without further pharmacokinetic studies requirements. Regarding the clinical trial performed on patients, Kanjinti was tested in HER2 positive early breast cancer, specifically a randomized double-blind phase 3 study in adult female receiving new adjuvant therapy, followed by after run-in chemotherapy, followed by surgery and adjuvant treatment up to 1 year. This is as shown in the study design details. Treatment sequence as is illustrated as first run-in, non-randomized. All patients receive doxorubicin, cyclophosphamide, 4 cycles for all patients. Then a new adjuvant. This is start for randomization. Paclitaxel, blinded trastuzumab, then surgery, as per investigator recommendation, and then followed by adjuvant. This is a blinded also trastuzumab, confirmed continued for 4 weeks to complete 1 year with optional endocrine therapy as per investigator recommendation. The key rationale for new adjuvant setting in HER2 plus APC provides a sensitive surrogate biosimilarity as trastuzumab enhanced pathologic complete response, allowing direct comparison of anti-tumor effect via short-term endpoint before long-term outcome aligned with EMA biosimilar guideline for oncology monoclonal antibodies. The endpoint measured is pathological complete response, and secondary endpoint is event-free survival, overall survival, safety, and immunogenicity. Our opinion for endpoint validity: PCR is well established, sensitive, and EMA-accepted endpoint in new adjuvant HER2 positive breast cancer. For a biosimilar confirmatory trial, PCR is appropriate because it is maximized sensitivity to detect a difference and it avoids unnecessary duplication of large outcome trials. For long-term endpoint, overall survival, safety, and immunogenicity, it is not required for established biosimilarity. Otherwise, this study is use this endpoint. The selected endpoint is valid, appropriate, and sufficient for biosimilar approval. Overall recommendation: it is recommended for approval by regulatory authority for comprehensive analytical similarity, pharmacokinetic equivalence, clinical efficacy equivalence, comparable safety and immunogenicity, and finally meet the totality of evidence standard required by EMA requirements for biosimilar. Additional data requested? No, there is no any additional efficacy or pharmacokinetic data required. Otherwise, routine post-marketing pharmacovigilance as per biosimilar standard. Thank you.
Okay, and now we are moving to part four, the part on the critical appraisal. First, my name is Asma Fouad, Head of Central Administration of Biological and Innovative Products and Clinical Trial at EDA Egypt. The first question was about the potential consequences in case of any differences in mode of administration or available information between Kanjinti and Herceptin in low resources setting. This may lead to medication error and incorrect preparation if administration instructions differ in infusion time, dilution volume, and staff rely on memory or outdated training. This may lead to wrong infusion rate or dose inaccuracies. These errors may increase toxicity or reduce effectiveness. The second result may be an increased safety risks, limited access to cardiac monitoring, trastuzumab as a trastuzumab risk, which means that any deviation in infusion procedure or pre-medication guidance may raise risk of infusion reactions or undetected cardiotoxicity. And the third result may be the it could be the compromised efficacy due to handling deviations if storage or handling instructions are misunderstood due to different labeling or missing leaflets. Consequences include temperature excursion, reduced biological activity, and patients receiving less potent doses. The second part of this question was a summary for the disagreement in the CHMP on the positive opinion of EMA. Of EMA. CHMP majority opinion dated on the 22nd of March 2018 supported approval based on the totality of evidences, analytical, non-clinical, PK safety, immunogenicity, and extrapolation, and concluded an overall biosimilarity of Kanjinti to Herceptin, while minority divergent position for three members of CHMP has signed on their disagreement based on the following justifications. The first is the clinical efficacy concern. Pivotal study, the number 2012-0283, did not meet pre-specified equivalence margins for PCR risk difference, so equivalence in efficacy was not statistically demonstrated. The second justification was the ADCC BCR extrapolation analysis linking low ADCC batches to PCR outcomes were inconclusive and too small and variable to confirm impact. And the last justification was due to the post hoc efficacy analysis rejected as approaches that excluded patients or altered covariates after the fact were considered biased and not acceptable. And the conclusion of the minority was the biosimilarity in efficacy was not demonstrated, therefore, authorization should not be granted. And the third part of this section was a question on the problem in biosimilar congeniality or the reference Herceptin for these variable outcomes of the clinical studies. The problem was not on the congeniality side. The variable outcomes were not considered to be caused by a problem in the congeniality itself, but were mainly linked to variability in some batches of the reference product, the European sourced Herceptin used in the comparative trial. The biosimilar still showed acceptable similarity overall, and the sponsor provided evidence that the differences were unlikely to be clinically meaningful for long-term endpoints. These are the disease-free survivals and the overall survival. The EMA CHMP majority still considered the benefit-risk positive and the approved congeniality. Now move to this part on the summary of the paper, Chemetol in monoclonal antibodies. The title of the paper was Drifts in the ADCC-related quality attributes of Herceptin, Impact on Development of a Trastuzumab Biosimilar. The paper was summarized in the three following points. The authors discussed lot-to-lot drifts changes over time in ADCC-related quality attributes of the reference product Herceptin. These drifts were linked to reduced or variable ADCC, the antibody-dependent cellular toxicity, activity in some Herceptin batches, and used those batches, unfortunately, was used during the biosimilar development period. This created a challenge for the biosimilar sponsor because the reference product was moving, making it harder to prove similarity on functional immune-mediated mechanisms like ADCC. The sponsor had to demonstrate that the biosimilar matches the current quality profile of the reference, not older drifting lots with expiry dates from August 2018 to December 2019. Based on this, CHMP reviewers questioned ADCC-BCR relationship, partially because low ADCC Herceptin lots may have influenced BCR variability, not the biosimilar itself. And based on this, EMA approval was still based on a totality of evidence, since ADCC drift was judged unlikely to change long-term clinical outcomes, which are DFS and OS. As a regulator, how could we deal with divergence data, grant or reject this MA? As a regulator, learning the lesson from such cases, totality of evidence is the base for the final regulatory decision. Also, evidence based responses from the sponsor of the biosimilar product made the regulatory decision more informed. The divergent data issue was resolved by knowledge and provided evidence on the lot to lot variability that's still in the accepted range and will not impact the long term endpoints. I will grant the MA based on the totality of evidences and the scientific responses submitted by the applicant to the authority. My opinion about applying the reliance principle in product assessment at EDA. Reliance principles are well applicable in this case based on transparency in the public assessment report together with the data provided from the local applicant to EDA. And Cangenti itself has been already granted marketing authorization in EDA through reliance mechanisms based on the sameness approach with complete CTD file and EPAR available in Egypt, and since 2018, informed reliance has been actively in place, which has been developed towards smarter and more dynamic regulatory processes that support patients' access to innovation and regulatory capacity building as well. And last but not least, lessons learned from this case. Biosimilarity assessment depends mainly on the stepwise approach and the totality of evidences provided by the applicant to the authority. Knowledge and understanding of the reference product variability is critical to regulatory decision. Functional differences and their impact on the clinically meaningful outcomes are also critical to biosimilarity development and conclusion. And last but not least, reliance always helps. Just make sure that it is an informed one.